Here in our first video of Module 3, we're going to look at conformational analysis, focusing on simple molecules to begin with. So here we have ethane drawn out with wedges and hash marks to show some of the geometry, right? Here's our eye point of view. So we're looking at it on end like this. And certainly building the models on this really helps. If we look at it on edge, we need a way to draw that, and we do that using the Newman projection. It's really useful to say where you want your projection to be viewed from. So I say from C1 to C2, from carbon 1 to carbon 2 here. If we look at it from carbon 1 to carbon 2, we have the front bonds attached to carbon 1 in blue. So it's color-coded here. The blue is in front, the green is in back. The convention is that you draw the larger greens, the larger circle in back. In this case, it's green. And to indicate that that is the carbon that's in the back. So this carbon's in the center. On the ends of all of these lines, you have to draw what's present, otherwise it would be implied. So had I not drawn this hydrogen, for example, and just left the line as it is, that would communicate to the reader that that's a methyl group there implied on the end of that line. So you have to specify what's there without shortcuts. Notice that we have this such that the bonds do not overlap. This is called a staggered conformation. So in a staggered conformation, the different substituents are rotated about as far apart as they can get. And that's typically lower energy. So staggered conformations, of which there are several, are lower in energy. And usually what that means is the molecule prefers to rotate into that position and stay there longer. Let's take a look at the rotations around bonds in ethane from this point of view, and from the point of view of energy. So notice as it rotates around, the energy drops and then goes up, drops and then goes up. All of the up portions, if you notice on the diagram on the right, which is more like a Newman projection, all of the high energy points are where the atoms overlap. And so those are not staggered conformations, they're what's called eclipsed conformations. So we have staggered, and the way we would draw eclipsed would be back carbon, just trying to get them consistently as close as you can to the front ones. But you need room, for example, to write in what's in each of these positions. In this case, hydrogens, right? So this is eclipsed conformation. And these are typically higher in energy. So if we look at the molecule butane, we can specify that we want to look at a Newman projection from C2 to C3. So viewing it along the C2 to C3 bond. Here's our point of view picture, just like in the last example. Wedges and hash marks shown in there, but here you have the methyl groups, if you want to look at them that way, attached to carbons 2 and 3. So the Newman projection of this has the blue atom, blue carbon number 2 in front. It's those 3 bonds. The green in back. And notice the methyls are as far apart as they can be. So This would be the lowest energy state of this molecule. And in fact, it's staggered, but remember there's more than one staggered conformation. So the lowest energy staggered conformation is called the anti-conformation. So there's two other staggered forms of this molecule where the methyls get closer together. And we'll take a look at that in a video also. Key to all of this is remembering that you have free rotation around single bonds. So wherever there's free rotation, there's the likelihood of different conformations. These are distinct from configurations which involve fixed directions of bonds off the carbon atoms, say for example, that would require breaking those bonds and switching things around and reattaching them to get to the other configuration. So conformation and configuration. Looking at butane, in the same way, we see that there are different high energy states. As you might guess, the highest of the high energy states are where the methyls overlap exactly with each other. And so if we come around, it's right there is the highest energy state. Then you have two other high energy eclipsed states where the methyl overlaps the hydrogens on the back carbon. The low energy states are all the staggered states, but the lowest is where the methyls are farthest apart. And we'll get to that right there's the lowest energy state. So you could slow this video down later if you want. There's two ways that energy is increased due to strain, what we refer to as strain. So steric strain is the example of where the two methyls in butane are eclipsed. These, if you built the model, and especially if you built a space-filling model that really showed electron density, these are really bumping into each other almost. So there's a lot of repulsion strain on these bonds. Okay, so so that we call steric strain. The word steric is used to describe anything that has to do with space occupancy, I guess you would say. How big atoms and groups of atoms and parts of molecules are in space, sterically. So steric strain puts a strain on these bonds and therefore is higher in energy. And torsional strain is another one. So they seem very related, and they kind of are. So to rotate this methyl in this direction causes what we call torsional strain, a resistance to going in that direction to get closer like they are here. That actually adds an additional amount of energy. So both lead to higher energy, both types of strain. A few other interesting points to make are, for example, if you have, say, this and this again for butane. This is staggered, but there's another staggered form if the back rotates around. And so if you were to do a similar thing like we were in the video, rotating around and just looking at the energy levels, these two would end up being the same energy level. They're both staggered. So how do we distinguish one from the other? So we introduced the term gauche interactions. this is similar to the term anti that we saw before. So this here is a gauche interaction, as is here. And so in talking about the different staggered forms, there's these gauche interactions, but they are absent in the staggered form that's lowest in energy, which is this one, H. And so that's where we had the term anti. So there's often several gauche or gauche forms. People call it gauche who haven't had French. So, but there's only one anti form, and all three are staggered. Another thing to point out is that clearly you can measure angles for anything and talk about the different angles. And discuss these. And then, but the other thing is that we need terms for the different relationships between, for example, what is the relationship between the front methyl and the back methyl? How do we say that without having to say the methyl on carbon 1 is such and such distance from the methyl in carbon 2? Or how do we say the methyl in carbon 1 is at a whatever it is 60-degree angle from the methyl on carbon 2? How do we say that's shorter? So we refer to the relationship in space, relationship between these, is as vicinal. It's an interesting word, and actually a word that comes in useful again in our spectroscopy unit coming up in not too distant future. So we'll talk about these methyls, for example, are vicinal to each other. They're on adjacent carbons. So this hydrogen is vicinal to this hydrogen, and so on and so on. So you can always use the word vicinal. It's a strangely useful term. I try to actually limit the number of terms that we focus on just to the most important ones. The question sometimes comes up is, what is the importance of this? So I have a very brief thing here. The molecule shown in the middle bound to this protein, which is called opsin. All of this is in the retinal, in the cells in the back of your retina, retinal cells. And this is cis-retinal that you saw on one of your worksheets or homeworks, I can't remember which. Cis-retinal from vitamin A in your diet, broken down into different form, binds into the protein opsin in the cells of your retina. When light hits cis-retinal, it breaks the pi bond momentarily and allows it to change conformations. So there's a conformational change here. Into transretinal. So technically this is also a configuration change, a conformational change that leads to a configuration change, because cis and trans, without light energy or UV light, particularly energy, these are fixed molecules. So, but it's not incorrect to say this is a conformational change. Notice what it does to the protein around it. The parts of the protein around it get all skewed in different directions. When that happens, if you look at the larger biology of this, here's the area we're looking at right now. When it undergoes conformational change to the trans, it begins this series of signals through proteins and enzymes, leading to a signal down the optic nerve. to the brain, obviously. So this is fundamental to the whole process of vision. This is how light that hits the retina, the cells in the back of your eye, how it tells your brain basically that you are seeing something in the light. Didn't mean to go that long on this, but anyway, this is our first module, and we'll talk more about cyclohexane rings and other things in module 3, video 2.